So I'm really excited to speak to all of you a little bit about my research on human milk. I want to start with disclosures. So these are my funding sources, for which I'm very grateful. And I'll start with personalized medicine. That was in my title. This is a concept, I mean, it's a buzzword. I think different people, it means different things to different people. I think in a clinical audience, a lot of people think about personalized medicine as, Lee introduced me, we started working together on cancer. So a cancer drug that's personalized to the particular genetic polymorphisms in a tumor, for example. Or if you talk to nutritionists, they might think of it as a personalized exercise regime that's optimized for you and your metabolism or your body type. But when I think of personalized medicine, I think of human milk. And this is a quote that I love by Cesar Victoria, published in The Lancet, which did a special breastfeeding series in 2016. And if you get inspired by the talk today, I'd encourage you to go here as a place to start in terms of the summary of the research and evidence around breastfeeding, not only from a health perspective, but also an economic and public health perspective. So Victoria wrote in this excellent series that human breast milk is not only a perfectly adapted nutritional supply for the infant, but probably the most specific personalized medicine that he or she is likely to receive, given at a time when gene expression is being fine-tuned for life. So I love this quote because it wraps up for me in one sentence a lot of what I do as a researcher. So this idea that the timing is really important. So I'm a DOHAD researcher, DOHAD meaning the developmental origins of health and disease. And that entire field is focused on the idea that what happens very early on in utero or in early infancy can really have a lasting impact on life, because this is when so many of our physiological systems are programmed. So our immune system, our metabolism, and so on. And that's the period of time when breastfeeding occurs. And it also refers to this fact that breast milk isn't just food. Of course, it is food, and it's very important food, the only food that infants are consuming early in life during this critical period of time. But it's so much more than food. It contains many other components, and we'll talk about some of those today. I start out a lot of my talks with this quote, but it's especially fitting today because Cesar Victoria was a recipient of a Gairdner Award in 2017. And I can actually still remember sitting on the couch in the morning, eating my oatmeal like I always do, reading the news, and seeing the announcement of the Gairdner awardees for that year. And scrolling through, there are about 5 or 6 of them each year. And I was, about to close the article. Cesar was listed last, and I almost spit out my oatmeal because it was so exciting to me that a breastfeeding researcher would have won an award of this caliber. And if you've been watching the session today, you heard an introduction about Gairdner. These are known as the mini Nobel Prizes. It's a big deal to win a Gairdner Award. And the fact that breastfeeding was recognized on that level was really exciting to me as a new researcher starting out in that area. So I was really excited, and Cesar Victoria was kind of a hero of mine. He really pioneered a lot of the epidemiology in the 70s and 80s that showed breastfeeding was so important for infant health. There had been a period of time before that when it was thought that infant formula, which was this kind of new scientific substitute for breast milk, was much better. And it was really his work that started the ball rolling of understanding that no, breast milk is personalized in a way that formula never could be. And he not only did research to show that, but also did a lot of work in policy and implementation to find ways to support breastfeeding. So not just stopping at making the discovery, but making an impact with that information. And so I really wanted to meet him, and I reached out to the Gairdner Foundation and said, how can I come to the awards? And they said, sure, it's open to anyone. So I flew to Toronto, and I got to meet Cesar Victoria, one of my research heroes, and it was an amazing experience. And I won't go into that story right now, but I know many trainees are watching and participating in the mentorship sessions tomorrow. So if you ask me, I'll tell you the story of how this turned out to be a $5 million moment. And we can discuss that at the sessions tomorrow. So with that, I'll sort of move on to the meat of my talk, which is the research and evidence around breastfeeding and human milk and why I'm so excited about it. So I talked about breast milk as personalized medicine. This is kind of one buzzword. We also talk about breastfeeding sometimes as a baby's first vaccine. There are actually countries where the vaccine record starts not with the baby's first vaccine shots, but with whether or not the baby got colostrum, because it's that important to immunity early on. Also in the title of my talk, I talk about breast milk as a superfood. So we'll get into this a little bit. Often you hear colostrum, which is the very first milk, referred to as the original liquid gold. And maybe a bit cliche, but I really like this. Making milk as a superpower. This is a pretty awesome thing that mothers are able to do. And it's important to understand how it all works. And that's what my research really focuses on. So starting with this superfood claim, this was a perspective published in Science a number of years ago and framed breast milk as nature's first functional food. And so I really like this perspective. And this is another quote from that article that I really love by David Mills, a human milk researcher. He says, "Milk is really a genius fluid that was outrageously understudied. If we could identify the components of human breast milk that are important, then we can understand the wisdom of milk and take advantage of them." So what does he really mean about taking advantage? Well, to put some numbers to this claim, these are global data on child mortality, so mortality of children under 5 years of age globally. And at the Institute for Health Metrics and Evaluation website, I generated this map myself. So they have all sorts of data available and you can play around with it, looking at, for example, cause of death. And so this is a map showing you the proportion of child deaths under 5 years of age per country that are attributable to suboptimal breastfeeding. And these are data from 2013. So you can see that, fortunately, in our part of the world, not too many child deaths are attributable to breastfeeding suboptimization. But in some areas of the world, up to, this is close to 20% in South Africa of childhood deaths under 5 years of age could be traced back to suboptimal breastfeeding. This is a country where if babies need to have formula, that formula is often made with water that is unclean, that might cause infections. The medical care there is not as good as we would have in other places. Infectious disease is a major cause of mortality, especially in infants. And you can see that in other areas of Africa, there are 10 to 15% of deaths. So that same Lancet series that I quoted Cesar Victoria earlier has estimated that improving breastfeeding practices around the world could save over 820,000 lives a year. So these are big stakes when we think about breastfeeding and how we could take advantage of the knowledge of breast milk, even if every baby can't be breastfed, because we know that's not always possible. If we can understand how breastfeeding and how breast milk supports infant health, then this is information we could use to support health in all babies. Now, of course, that's one extreme, childhood mortality. And you can see it's not so much a problem in our part of the world, but that doesn't mean breastfeeding is not important here too. So this is a handout from Manitoba Health, so right in our own backyard, talking about reasons to breastfeed your baby. And so there are many, including that it's better for the environment, it can save you money. And the one I will focus on today is that breastfed babies are healthier. So it's not just about childhood mortality, but thinking in our part of the world, breastfeeding has been associated with a 36% lower risk of sudden infant death syndrome, a 72% reduced risk of respiratory tract infections during infancy, a lower risk of type 1 diabetes, and also a 15 to 30% reduced risk of obesity later on in life, so adulthood or adolescence. So SIDS and infections, these are things that happen in infancy, but diabetes and obesity, these are lifelong conditions that can be tied back potentially to breastfeeding. And then one reason that wasn't on that pamphlet, because it's a bit old, is to do with the microbiome. And this is actually how I very first got interested in breastfeeding. So during my postdoc, I worked with a child study, which I'll tell you more about in a few moments, and did some of the first analyses of the infant gut microbiome from that cohort. We published a paper in the Canadian Medical Association Journal in 2013. It's really small here, but it says profiles by mode of delivery and diet. So we found that the microbiome of babies was different depending on whether they were born by C-section or vaginal delivery, and also by whether they were receiving formula or breast milk. And at the time I did this paper, a lot of people didn't even know what the microbiome was or why it was important. I hazard to guess that if we were all in a room today and I asked you to raise your hand if you've heard of the microbiome, I think all the hands would go up. It's pretty well known now that these bacteria that live in us and on us are really important. And it's becoming clear that early in life, breastfeeding is really important for shaping that community. So again, I won't tell the story today, but maybe something for the mentorship sessions tomorrow. This paper, which you can see made the cover of CMAJ and actually received an award for Paper of the Year from that journal, was initially rejected and not even reviewed by the journal. So that tells you a little something about perseverance and also the way that science attitudes can change over time. 5 years later, this paper, which I was not involved in, but comes from another birth cohort study, made the cover of Nature. And with a larger sample of infants and more advanced techniques, they replicated what we had seen, which is that breast milk, of all these different factors that might influence the microbiome, was far and away the most important factor shaping the infant microbiome. So that's this bar here. You can see that as we showed, how you were born is important, it's significant. Whether or not a mother's obese, whether the baby takes probiotics, all of these are important, but by a landslide, breast milk is the most important. So why is that? And why did I then go on to focus my career on human milk? Well, to understand that, we really have to start looking at the milk itself. And so, you're not intended to read this, but I think this picture shows you very clearly that there are many things in human breast milk that simply aren't in formula. And formula companies are working all the time to make formula better, make it closer to human milk, but it's kind of no competition, and it would be very hard to replicate. And one of the reasons for that is that human milk is dynamic. So it changes. It changes from day to night. So the milk that a mom makes in the morning is different from the milk that same mom makes later in the day. It also varies even within one feeding. So milk at the beginning of a feed is lower in fat, and then the fat content increases towards the end of a feed. And that's thought to help babies understand satiety. And it also varies across lactation. So as the baby grows and develops, the composition of milk changes over the months. And it's also very different among mothers. So different moms are making different compositions of milk. And you can see that all of these things would be very hard to replicate in a one-size-fits-all formula. Milk is also personalized. So we talked about milk being a superfood. As a nutrition, it's personalized. It matches to meet the needs of the infant over the course of lactation. And it's also very specific to the species. So, all mammals make milk. That's one of the defining features of mammals. But if you think about just two species, so cows and humans, a human baby spends a lot of its infancy period sleeping. This is when its brain is developing, whereas a baby calf, if you've ever seen one be born, sort of is born and right away wobbles and gets up and starts moving around, and its brain is pretty much fully formed when it's born. So because of those huge differences in the priorities of a baby human versus a baby calf, the milk composition is really different. So human milk, for example, has much higher levels of fatty acids that are important for brain development. So if you start feeding cow's milk to a human baby, you can see how there would be a mismatch. And then we talked about human milk as a personalized medicine. So milk is not just delivering nutrients, it's delivering optimized immunity to the baby while its own immune system develops. So a baby is born with a very immature immune system, and it's relying on its mother's milk to get things like antibodies to protect it from infection while its own immune system develops. And so this is another picture showing you a similar message, but you'll be able to read the words, which is that formula contains the main building blocks that a baby needs to grow, so the main macro and micronutrients, but it doesn't contain many of these other things, like I had mentioned antibodies. Milk also contains hormones and growth factors. And this picture, again, is a little out of date. It doesn't refer to the microbiome, but we now know that milk also contains live probiotic bacteria, as well as prebiotic oligosaccharides. So that's food for those bacteria. Milk even contains stem cells, which is pretty amazing, not a focus of my own work, but something I'm always happy to talk about, as well as food antigens. And I've got that in capitals there because that's a particular interest of mine lately, because I'm interested in food allergy development. And these food antigens that occur in mother's milk can be some of the first exposures that a baby receives to allergenic foods like peanuts or fish. And we're working to understand how that works and how it might be beneficial in preventing allergies. So I really think what's important is not only to look at these building blocks individually, which is kind of how they're shown in this picture. What I think that's how research has been done in the past. You get a microbiology expert looking at the bacteria, a stem cell expert looking at the stem cells. But what I think is missing there is that we really need to look at them all together. They occur together in milk, and they're probably interacting with each other. So this new kind of philosophy on milk, the field is shifting towards thinking of milk as a system of many different biological components. And it's not only a system in and of itself, but it's a system within a system. So milk has got all these components, but it exists within this mother-infant milk triad. So the idea that the mother and infant are connected through milk, and it's actually a bidirectional connection. And so we have to think of all three of these together and how they influence the microbiome and other body systems to really understand the wisdom of human milk and how it's functioning. And so an ideal platform in which to do this would be a study of many mothers and their infants with breast milk samples. And that actually is what the child study is. So I mentioned this to you earlier. This is where I did that first paper looking at the infant microbiome. And so it was there that I got interested in breast milk and realized that we had breast milk in the freezer from all these moms whose babies we had studied the microbiome. So I was able to use this platform to study breast milk. And so very briefly, the child study is a longitudinal cohort study in Canada recruited from 4 centers, one of which is right here in Manitoba. About 3,500 families are participating. They were recruited around 2010 at the time that their mothers were pregnant. And so we've been following them, their children, their whole families ever since. This is still an ongoing study. And it was developed to answer the question of how genes and the environment influence child health and development. I mentioned allergies earlier. That's one of the outcomes addressed in the study. So really trying to understand why allergies have increased in prevalence so much in the last couple of decades. And so we are looking at genetics. We know that these conditions do tend to run in families, but at the same time, we know that's not the whole answer because our genetics as a human race haven't changed in the last couple of decades. So what has changed is the environment. We're looking very closely at the indoor home environment where babies spend so much of their formative months and years. We're looking at things that aren't as typically thought of as part of the environment, like stress or infections, social factors. We're also looking at nutrition, the outdoor environment, so things like air pollution, then of course the microbiome, which is how I got initially linked to the child study. And so the way I've been using the child study to look at breastfeeding is as follows. So firstly, asking the question, is breast milk feeding? And you'll see in a moment why I called it breast milk feeding and not just breastfeeding. Is it related to these different outcomes later in life? So allergies, asthma, but also obesity, diabetes, cognitive development. In this large cohort of mothers and infants, we're able to start looking at those associations. And then if it is, because we have those breast milk samples, we can ask the question, well, what is it in human milk that is important and driving those associations? And so there are many components, some of them I mentioned earlier. And then even beneath that, we can start to say, okay, but what is it about the moms that affects the levels of these different factors in the milk? Is it something modifiable? Is it her diet, smoking habits? Is it something fixed like age, ethnicity, or genetics? Or other things like probiotics, antibiotics. And we've collected information about all of these things in the child study. It's important to account for those because they might influence milk, but they might also simply influence a mother's ability or decision to breastfeed. And they might influence child health through mechanisms that are totally unrelated to breastfeeding. We can go one step further then if we identify milk components that are important and start to think about the mechanism by which they act in the baby. So is it through the gut microbiome, which was my initial doorway into human milk research, or is it a different mechanism, epigenetics, metabolism, and so on? And then my goal at the end of the day is not just to observe this in the child study cohort, but to use this information to develop interventions where we could support the next generation of mothers and babies, either to enhance breastfeeding, maybe to optimize milk composition, or to address some of those underlying factors. And then, there's lots we can do in human studies, but we can't do everything. So I do also collaborate with researchers who develop animal models to look at mechanisms and also to look at population level data. So there would be lots to say if I were to talk about all of this today. So I'll just give a few examples touching on these elements that I've highlighted in red. So the first being the connection of breast milk feeding with clinical outcomes. So in the child study, we asked a lot of questions of our families about how they're feeding their babies. And we asked it at repeated time points. A lot of longitudinal studies in the past have simply asked, did you breastfeed your baby? Yes or no? And maybe how long? But we asked a lot more questions than that. And, I won't ask you to read these, but just to give you the sense that we asked about, were they using bottles? Were they glass bottles, plastic bottles? Were they using formula? What kind of formula? And one particular question that has turned out to be really interesting is that we asked if mothers were using a pump to express their breast milk. And I wanted to point out that this actually wasn't my idea. I know that we have a lot of trainees watching today. And so Dr. Annika Klopp was a trainee, and she approached me saying, Megan, I see you're researching breast milk. I've been doing my residency in the NICU, and I see that these preterm babies are getting their mother's milk, but they get it through a tube or through a bottle. And does that make a difference? And I had honestly never thought of that. And I didn't even know that we had the data, but we looked back through the questionnaires and found this question. So we were able to look at that. And so this is one example of a study that we did together where we looked at the infants in the child study and classified them according to how they were fed. So whether they received breast milk only, all directly fed at the mother's breast, whether they received all breast milk, but some of it pumped and fed indirectly from a bottle, or whether they received formula or some combination. And you could see here that looking at asthma at 3 years of age, it was the formula-fed babies that had the highest rate, the directly breastfed babies that had the lowest rate, and the babies receiving some pump milk were somewhere in between. And to us, that was really interesting. It's not what I expected to find, but it's what the data showed. And so our next question, of course, was why? Why would that be different? And we had a lot of ideas, some of which we're still exploring, but it could be that the bioactivity of the milk is affected by the process of pumping and storage. Maybe the microbes in the milk were affected. It could be that the skin-to-skin contact between a mother and baby is important. And of course, that doesn't happen if the milk is pumped and fed from a bottle. It could be about contaminants in the bottle, the plastics. It could also be this infant-mother signaling that occurs. So these are still things we're exploring, but it's an example of the kind of hypotheses we can generate with this type of research. I do want to make my standard PSA here about pumping. Unfortunately, some of this research has been reported in the news in a way that I don't think is fair or helpful. Talking about pumped breast milk containing nasty bacteria. This really isn't the message that I think should be conveyed, but I do think it shows that pumped milk is not equivalent to nursing at the breast. However, we do see that it's beneficial compared to formula, so that's a good thing. We also know from other research that it might be the only way that a mom can provide her own milk to her baby for a variety of reasons. And so that's really important to recognize. And at the end of the day, it's important to understand why. So if we find that it's something about the way the milk is stored, we can then advise moms to store the milk differently, and so on. So moving on then to talk about the milk itself. You can see here, this is an updated version of the figure I showed earlier, because it does include those pre and probiotics. And I'm slowly chipping away at these blocks and getting them all analyzed in the child study with that goal of looking at them all together as a system eventually. So some of this work is already published. Some of it is underway or coming soon. And I'm always looking for new collaborators who measure other things. My wish list is always growing. What I'll focus on today is a little bit of data on oligosaccharides, which I have done in collaboration with Lars Bodie at UC San Diego. So human milk oligosaccharides, I mean, it's hard to pick favorites, but if I had to, I think these are one of the most important and exciting components of human milk. So if you think about milk, it's about 90% water. And of the solid components, the most abundant one is lactose, followed by lipids or fats. And then even outweighing proteins is human milk oligosaccharides. And so these are non-digestible carbohydrates, meaning they're not food for the baby. The baby doesn't digest them, but we now know they're food for the baby's microbes. And we also know that they're structurally diverse and very species specific. So for example, cows make milk oligosaccharides, but they make about 40 different kinds, whereas humans make over 100. So as soon as I heard this, I thought, well, that's amazing. Like, this is one of the reasons that human milk is so optimized to human babies. And, even though this was known, there are many different human milk oligosaccharides, there really wasn't that much known about why there are so many different ones. What do they all do? Why are they so different between different moms? And so I approached Lars Bodie as one of the world experts in studying milk oligosaccharides and said, hey, I have 2,800 breast milk samples in the freezer. We should analyze HMOs. And so we did. And the other thing that wasn't known is, okay, they're feeding the gut microbes, but what health impact does this have on the babies? And so this is something we're still actively exploring, but to show you a little bit of data. So this is Lars and my postdoc Cosetta, who worked hard on this data. Here you're seeing from about 430 moms in the child study that we analyzed the human milk oligosaccharide profiles. And you can see that all the different colors are different HMOs. From the mom who makes the least amount to the mom who makes the most, there's almost a 4-fold difference, so a huge variation. And if you look at the individual HMOs, there's up to a 100-fold variation between moms. So we're still working to understand why this is. One thing we do know is that secretor status, which is a genetic trait, and you can see down here, I've separated the moms into non-secretors versus secretors. This is a single polymorphism difference, is one thing that separates moms into these kind of two clear groups, but there's still a lot of variation between them. The one thing you will notice as the main driver is that this blue oligosaccharide, 2-fucosyl-lactose, is only made by secretors. And interestingly, this is one of the components that formula companies are starting to add to their products to make them more like human milk. And they've chosen to add 2-fucosyl-lactose, this blue HMO that is only made by secretors. And so I think, it's an interesting question to ask. If about 25% of moms are non-secretors and do not make this HMO, is it a good thing to put it in formula and give it to every single baby? I think we don't know the answer to that. Now, is this linked to health outcomes? Well, one thing we did in the child study was to link the HMO data with food sensitization, an early form of food allergy. And what we saw was when we looked at HMOs one by one, these are little tiny graphs, but all you need to know is there was no association. We didn't find that any particular HMO was different in babies who remained healthy or who developed allergies. But when we looked at all the HMOs together using a machine learning method, we saw an important difference. And so to me, this signals that there's no single HMO that's going to predict food sensitization, but the overall profile is important. And that's just looking at one component. We still don't know the mechanism. That's something we want to learn. What happens when you add other components to this analysis, right? And think of milk as a system. And what about other conditions? So food sensitization is just one, but we could easily look at asthma and obesity and cognitive development. So there's a lot of research yet to be done. I touched on these few red points. And I mentioned that we eventually want to look at interventions. And that's something that we have to do beyond the child study because child is an observational cohort. The kids are now 10 years old. If we want to really develop and test interventions, we have to think of new ways. And so I want to briefly tell you about two other programs in my lab. The first is MILK, the Manitoba Interdisciplinary Lactation Center, funded by Research Manitoba and the Canadian Foundation for Innovation. This is a photo from the day it was announced, and the Minister of Health and Science came to share in that celebration. And the other is IMIC, the International Milk Composition Consortium, which is funded by the Bill and Melinda Gates Foundation. And it was an exciting day for me when that was announced. This is a snapshot from the front page of the Free Press, which I know I've never been on the front page of the Free Press before. And I'm pretty sure it's safe to say breastfeeding has never been on the front page of the paper before. So I think that was a really exciting day for multiple reasons. So starting with IMIC. This study, as I mentioned, is funded by the Bill and Melinda Gates Foundation, and it speaks to that intervention piece. So the foundation had already been funding 4 randomized trials of different nutrient supplementations for mothers, and they saw some of my talks and other research and said, it's pretty clear that milk is an important piece of this puzzle. So they approached me and said, if we get our studies to collect milk samples, then would you analyze them and try and understand whether the milk is changing in response to these interventions? And if so, is that important for the infant outcomes? So we connected with these ongoing studies. We're analyzing data and samples from about 1,000 mother-infant dyads. And we're looking at all the different components of milk. And then we're working with some data scientists because when you generate these many data sets, it's not straightforward to analyze those data. So we're working with them to understand how these different milk components interact and how they influence different health outcomes. And so these data science partners are from Stanford and Berkeley, and it's been super exciting to work with them and apply these advanced methods to the analysis of milk data. And you can see that in the IMIC study, I'm getting close to the full rainbow of milk components. So it's really exciting. And the last thing to touch on is MILK, the Interdisciplinary Lactation Center here in Manitoba, which I co-direct with Dr. Nathan Nickel. Nathan comes from a public health sciences and biostatistics background and works at the Center for Health Policy. And then I have a background in human milk and epidemiology. So we're really complementary. And we're really excited about the center, which involves 3 main pillars. One is a database of information about infant feeding that Nathan is leading. And here is where we're collecting feeding information from all infants in Manitoba is the goal. We're getting there step by step. And then a human milk biorepository, which I'm leading, where we have freezers and milk analyzers and equipment to analyze human milk. And so the idea is to build up a repository of milk samples that will then be linked to the feeding data and to the population research data repository at the Center for Health Policy, so that we'll be able to understand not only how human milk is linked to maternal and infant health, but also how different population level interventions or policies are changing breastfeeding rates. So I know I'm close on time, so I'll zip through the next little bit, which is about the feeding database, which really allows many unique possibilities. We have several thousand infants already in there. Our biorepository, which is really exciting, up and running. It was born during the pandemic last April, which has been an interesting challenge this year. And then we're also, through milk, working on some clinical trials where we're looking at, for example, the effects of different milk fortifiers on the microbiome of preterm infants in collaboration with one of the neonatologists at Health Sciences Center. We have another trial where we're looking at ways of matching human donor milk better between mothers and babies, which I'd be happy to talk more about if people have questions. And then very importantly, we have Milk Club, which is a monthly meeting of researchers, trainees, community health workers, clinicians, and policymakers who are all interested in breastfeeding and human milk from different perspectives. And so we work collaboratively to do things like develop new studies, recruit research participants, and share evidence. And on that note of sharing evidence, this is the final thing I wanted to touch on, which is that I think it's really important not only to do the research, but to get it out there to the people who need it. So I do things like speak to the media. I've been a part of developing some courses for healthcare providers for them to learn about breast milk and the microbiome. I was part of a committee commissioned by the National Academies of Sciences, Engineering, and Medicine to scan the evidence on human milk and use that to develop nutrition guidelines for infants. I've developed some animated videos targeted to the public audience to get them understanding and appreciating the amazingness of human milk. And some members of my lab have been involved in writing commentaries, for example, about breastfeeding during the pandemic. Oh, I lied. I have one more exciting thing to tell you. So I focus mostly on work going on in Manitoba, but if you weren't convinced yet from that breast milk is a superfood and a personalized medicine, I have a couple quick stories for you. So this was the quote we had earlier, superfood for babies, but it's not just about babies. So there is some research going on looking at, for example, how human milk could help people recover quicker after a bone marrow transplant because it can support the immune system. So this is done in adults. There's also studies going on looking at HMOs and how they might help in treating irritable bowel syndrome, again, in adults. So we can learn a lot from human milk and apply it well beyond infants. And then, this is an example I had to give because of what we're all experiencing right now. So breast milk as a vaccine with antibodies, there's been a lot of interest in whether COVID antibodies are found in breast milk. And they are, not only in women who've recovered from COVID, but also in women once they're vaccinated. So babies, of course, are too young to get vaccinated themselves. But if their mom's been vaccinated, she's providing those protective antibodies to her baby. And so the WHO is therefore strongly recommending breastfeeding even during a pandemic. And there actually are studies ongoing to see if breast milk could be studied as a potential treatment for COVID-19. So lots of exciting research in this space, and it's changing almost on a daily basis. All right, so I hope that many of you out there are inspired to learn about human milk. If you are, there's an international society that you should think about joining. They have fabulous programs. There's a conference coming up this summer. And in particular, they have awards for trainees up to $10,000 to do a research exchange, go visit another lab, could be mine, but doesn't have to be, to learn about human milk science. So with that, I want to thank my collaborators, my team. Everything that I showed you today has been a huge collaboration, team effort. These studies take input from a lot of people. My collaborators at Milk and the Child Study in particular, and all of our funders. And I would be happy to take questions.